Hi, and thank you for joining us for Chardan's Virtual Ophthalmology Summit. The focus of this installment is on rare ophthalmic diseases. My name is Daniil Gataulin. I'm one of the senior biotech analysts here at Chardan. It is my pleasure to introduce George Magrath, the CEO of Opus Genetics. George, welcome. Thank you for joining.
Daniil, thank you for having us.
Of course. So the format that we are following for the summit is the fireside chat. So I'll have a number of questions to jump in and go through the company history and some of your key programs. So with that, George, if you can start by spending a few minutes to introduce yourself and introduce your company and some of its key programs to the audience.
Yeah, of course. So I'm the CEO of Opus Genetics. I'm an ophthalmologist by training. I trained in Philadelphia in rare retinal diseases, and it's been an honor to be at Opus. I've been here for almost 2 years now, and we have a great team that's dedicated towards the next generation of gene therapies out of the University of Pennsylvania and others universities as well. Really on the backbone of the work that Dr. Gene Bennett did, the inventor of the first approved genetic medicine, which was Luxturna at Spark Therapeutics. And so we have a portfolio of 7 of those that are aimed towards 7 genetic defects where there's a core principle of structure function dissociation, where the photoreceptors, the anatomy is all present, but there's one ingredient that's missing. And so we just try to augment or add back in that one ingredient. And so it's a very straightforward type of gene therapy.
Got it. Excellent. So let's jump into your programs right away. So you have a couple that are more advanced. The most advanced is the LCA5, Leber
Mhm.
congenital amaurosis 5. What can you tell us about this disease? How is it different from other types of LCA?
Yeah.
And perhaps if you could give us an idea of what the addressable market is.
Yeah, boy. Well, LCA5 is one of the most severe of these inherited retinal degenerations. These babies are typically born and lose vision pretty early in life. So it's usually diagnosed in the first year or two of life. Like many of the retinal degenerations, the mom notices that the child can't see to pick up Cheerios or toys, things like that. And they pretty quickly get a genetic panel that diagnoses LCA5, which is the absence of a structural protein in the photoreceptors called lebercilin. And so those photoreceptors are still present. They just don't have the correct structure to allow them to sense light. And what we do with LCA5 gene therapy is replace that lebercilin to give that structural protein back to the photoreceptors. In the United States, there are about 200 of these patients, 57 of which are treated at the University of Pennsylvania. So pretty good concentration of the patients there. And fortunately, Dr. Tomas Alleman is the principal investigator on our study at the University of Pennsylvania. So it's a really it's an ultra rare disease, but it's a disease where we can have a dramatic impact in patients.
Got it. Okay. So there's another type, LCA4,
Mhm.
that is also quite severe, if not more severe than LCA5. In
Mhm.
LCA4, I think most patients retina degenerates by about 4 years. I understand that's
Mhm.
not quite the case with LCA5. Right, there's potentially a wider therapeutic window for those patients because the rate of degeneration is slower. Am I right about this? Can you tell us how quickly or
It, exactly. So one of the reasons Dr. Bennett picked LCA5 as a therapeutic target was because it retains the structure for quite some time. So those photoreceptors aren't present, are still present. So one of the problems you get in some of these diseases is that the photoreceptors completely atrophy. If they completely degenerate, well, then a gene therapy is not going to help because there's nothing for the gene to replace. At that point, you need some sort of cell therapy or optogenetic type target. But with LCA5, those photoreceptors can stay for decades. And so what it does is it allows you to have a structural protein or a structural cell that just isn't functioning. And so it's a from when you think about it from a therapeutic intervention standpoint, this idea of structure function dissociation. So the structure has not yet deteriorated, but the function's not there. Allow is sort of the perfect disease state for a gene therapy because you have a wide therapeutic window.
Okay, and when you say wide, what you mentioned it could be a decade, so you're not
Mhm.
limited to,
Exactly.
patients.
Yeah.
Uh,
So the our first patient that we treated in LCA5 was he was in his late 20s. This is a person who never had formed vision, right? He could see kind of blobs and shadows his entire
mhm.
life. And he was able to get back enough vision where he could read on the eye chart in his late
Well,
20s. And so, I mean, it was kind of cool. Like I've never really been part of a trial where you have to actually teach the patient what letters look like because they had never seen letters before. And he talks about things like being able to see his food on his plate stuff. It was very, very cool to his experience, really transformational. But that patient, like I said, was in his late 20s. And hopefully we treat patients earlier than that, but it gives you an idea of how wide the therapy window can be for some patients.
okay, got it, understand. So in terms of the construct itself, so the therapy
Hmm.
is called OPGX LCA5,
Hmm.
and it encodes, as you mentioned, libercillin. So
Hmm.
what is that libercillin? What is its function in LCA5?
Hmm. Lebercilin is a structural protein, and so it when in a normal photoreceptor, it's elongated, right? So it's a long, skinny cell that has that has the visual pigment in it, and then when the visual, when light hits it, the wavelength of light, it changes the configuration of those molecules, which creates electrical impulse that goes to the brain, right? And that's the 10-second view of the biochemistry, but without lebercillin, that long, skinny photoreceptor kind of coils down, almost like a spring. And so instead of being elongated and straight, it's more like an accordion. And
Mhm.
so when you replace lebercillin, it gets that long, skinny form again. And so that's exactly what we're trying to do. So it's a structural protein that has to do with the microtubule formation in the cytoskeleton of the photoreceptor.
Okay, got to understand. And in terms of additional features of the construct, what AAV type is it, and how is it administered?
Yeah, so it's all of our AEVs are either AV2 or 8 for our programs. This one's 2 because we're trying to select a tropism for photoreceptor, and it's a pretty specific vector for transfecting photoreceptors. It's delivered through the subretinal space, so it's a subretinal injection, just like Luxturna,
Mhm.
that's been popularized by that one. And so what you really are doing is you're giving 300 microliters of fluid to subretinal space, which is a great thing in the eye, and that this is not unique to us, but for being in ophthalmology is wonderful because the systemic or off-target effects are much simpler to deal with than they can be for systemic administrations. Our volumes are less, our CMC is a little less, our the off-target transfection is a little less because it's so targeted.
Right, makes sense. Okay, and in terms, it's in the clinic now. But before we
Mhm.
talk about the clinical trials and the early observations that you have from the clinical study, what preclinical data jumped out at you that give you the confidence in that program?
Yeah. So there's a great preclinical model that was done at the University of Pennsylvania that really showed improvement of the electrical potential of the eye when you deliver this. And so that's what really excited us was that the improvements were on the same scale as what was seen with, say, a Luxturna or other products that are starting to pan out. as actually being beneficial to patients.
Got it. Okay, makes sense. So let's shift gears and talk about the clinical program. So as
Mhm.
I mentioned, it's in the phase 1/2 study.
Mhm.
Um, can you tell us about the design? How many patients and what baseline characteristics those patients have?
Yeah, yeah, exactly. So like you said, it's a phase 1, 2 study, dose escalation study. And the idea is you treat 3 adults and then 3 children for each dose, right? So we've treated the 3 adults and we now have about 18 months of follow-up on those patients. And we've treated the first 3 kids. And that's the data we'll be reporting in the third quarter of this year. So imminently will be the first pediatric data. And the rationale for this is that the adults should be more in stage. Now, I already told you about the first adult, which had a dramatic improvement in his quality of life. The second 2 adults were a little bit younger. The second patient was patient labeled 01, um, which was a early 20s. And then the third patient was labeled 04, and that was a 19-year-old. So you can see we're starting to treat younger and younger, trying to get to um, as modifiable this disease as possible. And that's exactly what we saw was that the efficacy is um, is enhanced as you go younger and younger. And so we're excited to see what potentially could happen in these adolescents that we're treating now. And so the idea, is we got RMAT designation on the first 3 patients, which to my knowledge is the lowest number of clinical data, lowest number of patients that have been awarded the RMAT designation at the FDA. But it's allowed us to be very collaborative with the FDA on what's next. And so we're constantly sort of talking to the FDA about, okay, how do we dose escalate? What does a pivotal look like? What are the endpoints you care about? So it's an ongoing discussion.
Got it. Excellent. So you mentioned there's only about 200 patients or so in the U.S. Does and I guess a big portion of them are identified and being treated at, I think you said University of
Yeah.
Pennsylvania, right?
Yeah.
Um, do you foresee any challenges in recruitment given that small population size, or because all of these patients or majority of them have been identified, they're very willing to go?
No, we think we should be able to find these patients for a number of reasons. One is because we are very close with the University of Pennsylvania, and they do have a lot of these patients. But as importantly, we're really tied in with patient advocacy groups, including the Foundation Fighting Blindness, which is an amazing organization run by Jason Menzo. And then the LCA5 patient advocacy group, we were there about a month ago at their meeting. And so we're really tied into these patient advocacy groups. And it shows, right? The amount of non-dilutive funding we've gotten for our programs is in my mind, is quite extraordinary. I mean, every one of our 7 programs has some sort of ancillary funding, and it allows us to really do this ultra rare development.
Got it. Okay. And so you presented data from those 3 adult patients
Mhm.
from treated at low dose. Can you walk us through some of the key observations, if there were any safety signals first, and then perhaps some of the efficacy signals that you were excited to see? You already alluded to one patient, but if you can summarize the data for all 3.
Yeah, of course, Daniil. I mean, it's such an exciting data set, right? It's the first data set I've been a part of where on the first 3 patients, the FDA actually gave us a grant for the rest of the study. So the FDA is actually writing checks to support this study, which is very unique through the Orphan Products Division. And so the data, the safety part to start there, it's very well tolerated. It has the same sort of safety profile as, say, a Luxturna does with subretinal injection. So that part so far has gone well. From an efficacy side, the first thing we looked at was called FST, and FST is full field stimulus threshold, And that tests the overall electrical sensitivity of the eye. And so you shine a bright light at the patient, and they click a button when they see it, and then you just vary the intensity of that light. And that was the first endpoint that moved in Luxturna. So in these Luxturna patients, the very first endpoint was about a log and a half improvement in FST. And so for our first three patients, that's exactly what we saw, was somewhere around a log and a half improvement in those three patients. So that got us really excited. So the next thing we looked at was the maze, and this was the approval endpoint for Luxturna. Well, the maze, since Luxturna was formally approved in 2017, it's had a couple new iterations. It's now done in virtual reality using like a Meta headset. You put the headset on and you tag objects as you walk through a virtual reality maze. And so what you see in our slide deck is really for the two patients that were able to have vision enough to see. They were able to tag more objects at each luminance after treatment than they were at baseline. And then after that, pupillometry is another interesting endpoint because this is completely objective, right? You're measuring what the pupil does when you shine light. And so in a normal person, if it's dark, your eyes dilate, and then you shine a light and it constricts, and it's completely involuntary. So it's a nice objective endpoint. And we saw improvements in that as well. So you can
Mhm.
see everything is sort of going the right direction. And then lastly, there was one patient who was able to do microperimetry, which is point-wise testing of the retinal sensitivity in exact sort of points. You create a grid for it. And that patient had about an 18-fold improvement in that, which is astonishing. BCVA changed when compared to baseline and compared to the fellow eye. And we had a KOL day about in December where one of the KOLs, Christine K, her one of the things that stuck with me from that was she said it's unusual to see a study where this many endpoints all go the same direction. And that I think is the totality of our data where everything is sort of going in the right direction.
Okay, got it. And you mentioned that there could potentially be a correlation between the age at which those patients get treated and the effect that is observed. Were there any other predictive factors, or are you looking at any other potential predictive factors that could inform you which patients would be better responders?
Yeah, it's a good question. So we certainly think age is related, but the reason age is related, there's two reasons age is related. And the one is because we're looking for patients to still have the photoreceptors present. So what we don't want is we don't want atrophic patients. And so if you select out patients where the photoreceptors there, it's just missing lebercilin, then you got the best chance of success, right? Best positive probability. The second thing for kids is that you want kids who are still developing their visual cortical pathways. And so once you're an adult, we kind of get like a little bit sort of set in our ways as far as neural pathways go. But in kids, they're kind of constantly forming, changing, adapting, really a lot of plasticity there. And so we think kids not only will have an improvement in their retina, but also hopefully, fingers crossed, they'll have an improvement in the visual pathways that form if they actually have vision. And so I think if you can get those pathways set in childhood, you got a good chance of making the biggest impact on a kid's life.
Okay, yes, makes sense. Okay. And in terms of the dynamics of effect, at what point after treatment do patients begin to observe benefits?
About 3 months. Yeah.
But
So in the three adults, we saw improvement at 3 months, probably peaks at 6 to 12 months, and then is sustainable out past that to 18 months.
okay, okay. And you also alluded to one of the endpoints that you're looking at is the mobility test. It's a slightly different version than what was
Mhm.
used for Luxturna. It's called multi-luminance orientation and mobility
Mhm.
test. Uh, so what are the key differences between those? That's one question. And then is that a new standard? Are other programs relying on this test now, or is that specific to your programs?
No, I think it's becoming the new standard for a number of reasons. And that gets to the first part of your question. It's done in virtual reality, and so it's much more controllable. And so Dr. Bennett developed both these tests, right? She developed the one for Luxturna, and she developed this one. The old one took a big room. You had like movie set type lighting to try to really control the luminance, and it took about a full day to do. This one, you slip the headset on, everything's reproducible. You can put it at sites across the world. It's not, it's much more user friendly. And additionally to that, you can collect a lot more data because you have those cameras inside this thing and you have the computer that is collecting just tons of data. I mean, everything you could think of. How they're moving their hands as they go through, how fast they're walking, essentially, where their pupil gaze is, things like that. So you can collect a lot more data than you can with the original maze. And honestly, it's a lot safer. I mean, it's not the best ethical idea to put blind people in a dark room and then videotape them tripping over objects. I mean, that's what the original maze was. So I'd much rather do that in virtual reality than I would in real life. So, so it is becoming the new standard. Dr. Bennett's done a lot of work, published and unpublished work on 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 correlating this back to the original maze, and it's available for any program. And so I would encourage anybody else in the field that's listening to this to consider Dr. Bennett's newer version of the maze for their program as well.
Got it. Okay, noted. And so, one last question, and
Mhm.
for LCA5, what are the next steps and the timing for next steps? How are you thinking about the next cohorts and then potential transition to pivotal?
Yeah, so we're using it's super cool. We're using a data-driven adaptive design to this protocol, and we're working in conjunction with the FDA on next steps for all of this. And so in the very near future, Q3, we're going to be announcing the pediatric data, the early pediatric data. And when we announce that data, we'll be taking that data to the FDA to talk to them about, okay, what does a pivotal trial look like? How many patients should we enroll? What endpoints should we look at? Things like that. And so it's assuming the data is positive. If the data shows a little bit to be desired, then we dose escalate, and we're still a log unit below Luxturna, so we do have room to dose escalate. And so that sort of is the adaptive design, is that as soon as we see efficacy that's meaningful for patients and that we think is appropriate for what this therapy can do, then we're going to flip it into a pivotal trial.
Got it. Okay, makes sense. All right, let's shift gears, talk about your other program, Best
Mhm.
One. It's a type of bestrophinopathy. Can you tell us a little bit about that disease and how patients manifest and just in general disease pathology, any background you can share?
Well, this is like from a pathophysiologic standpoint and from a biochemical standpoint, it's a super cool disease, because you end up losing a calcium chloride channel that that allows fluid to pull under the retina. So you lose the calcium chloride channel that actively pumps the fluid out, and when you don't have that, the fluid backs up. So what are we doing? We're just replacing that channel. And so it's a very, very elegant, simple thing. And we would think that when you replace that channel, you should be able to pump the fluid out. So these patients, it's a horrible disease for these patients, right? They get the fluid under the retina, and it starts off with some distortion in their vision as the photoreceptors are now detached. And over time, a detached photoreceptor becomes atrophic. And so if you can basically empty that fluid before those photoreceptors become atrophic, you can modify their disease and you should be able to restore their vision. And so our in a dog model that was published in PNAS that was done again at the University of Pennsylvania, um, that's exactly what they saw was on imaging of the eye, on an image called an OCT of the eye. You can see that material or that fluid under the retina. You treat them with the gene therapy, that fluid just resorbs and it goes down flat. So from a clinical trialist standpoint, we're going to know about target engagement pretty quickly with this because we'll see, hopefully, in humans, the same response where that fluid just goes away.
Okay, got it. And in terms of the potential addressable population, how does it compare to LCA5? Bigger,
It's
smaller?
Yeah, it's much bigger. So if LC5 is 200, this is like 9,000. So it's a much more common disease.
Okay. And similarly, is that also, given the nature and the mutation, is that um, something that would benefit much more from early administration, correct?
Yes, yes, we think so. Yeah. Particularly in the recessive patients that have none of the calcium chloride channel, those patients tend to have fulminant advanced disease really early in life. The dominant patients, it can be later in life, but again, it's just like with like wet AMD or some of these other diseases. Like the less fluid you have in the retina, the better the patient does.
Hmm. Okay, yeah, makes sense. Okay. So in terms of clinical development plans, I understand that this program is still in IND enabling studies, correct? But you're
Mhm.
getting ready,
We're
but you're
getting
getting
ready to
ready to.
Yeah, so in the second half of this year, we're going to dose the first patients, and
Hmm.
we should have early imaging data, OCT data, in Q1 of 26. And so we're hoping to be able to show a resolution of that fluid in humans to match the animal data by by the first quarter of 26.
Okay, got it. And then mechanistically, you would expect the concurrent improvement in function as well, right? So you would have fluid resolution, and then at the same time or shortly the day after, there should be improvement in vision.
Exactly. So what you see in the natural history studies is when you do that microperimetry test that I talked a little bit about in LC5, you can do
Hmm.
point-wise kind of mapped out sensitivity of the retina. You see that when the retina is detached with that fluid under it, you start to lose sensitivity of the retina. And so we would expect that to change first. So after that retina flattens out, those points that are now in a more healthy position should regain sensitivity.
Okay, got it. And initially, it will be a standard dose escalation study, right? You start with three patients, 3 by 3 design, correct?
Exactly. Hmm.
Awesome. Okay. All right. Yeah. So we definitely look forward to updates on that program and
Hmm.
potential data in the Q1 of next year. In the next couple of minutes, if you could give us an overview of some of the other IRD programs that are next in line to advance, and which ones are you more excited about? How do you prioritize among them?
Yeah, Daniil, it's a great question. It goes back a lot to the non-dilutive funding that we spoke about earlier, because these programs that are preclinical, that's how we sort of prioritize them. We've picked out a bucket of these that were hand selected by Dr. Bennett and Dr. Yerxa too, who was a co-founder of the company, and those are all we feel very confident that all of those have a good shot on goal for being effective. They're just straightforward, AAVs, subretinal, looking at structure function association. So with that being the case, with the science being good, we prioritize them by our ability to finance them through non-dilutive measures, right? And so my general philosophy is I try to use equity dollars to develop clinical data, right? Because that has the most bang for the buck. And then the non-dilutive funding we target towards getting these things ready for clinic. And so you'll see that recently we announced a deal with MERTK, where we got $2 million for the IND enabling of that one. We're super excited about that one, and there's a number of reasons that we'll announce in the future about that program that it gets quite interesting. Um, there there are a number of others as well. Each of those seven has some sort of financing that should help bring it to the clinic. So we're excited. I mean, internally we call it a string of pearls, right? We expect to have 1 to 2 INDs each year, and these things should just roll one right after the other.
Okay. And is that something that you think could potentially benefit from the FDA's new platform designation?
Yeah, well, we're definitely looking into that, right? I mean, we're obviously close to the FDA, right? I mean, they're funding one of our programs, and so we stay in close contact with them, and so there are a number of things we're discussing, and that platform sort of program is definitely one of
Got it. Okay. So also given the number of potential new indications that may be online in the next few years, are you thinking of incorporating potentially new AAV vectors, the next gen, so to speak, vectors to improve upon delivery?
So we're always considering it, and we're always looking at it. But one thing I want to be real clear about is that at our company, I view this more as drug development, not drug discovery. And so I want to do things that have high likelihoods of success and have low capital requirements. So things that are early and have been unvetted tend to cost more to manufacture, tend to have a higher failure rate for manufacturing, can have a higher failure rate for clinic. And so we're always looking for a new, better mousetrap, but we are very disciplined about execution and being able to, I mean, at the end of the day, what generates value for shareholders is positive clinical data for a drug that's improved the life of a patient, because that's what will end up getting reimbursed. And so that's what we're after.
Okay, got it. Yeah, that makes sense. All right. And a couple more questions. In terms of CMC, how are you thinking about the CMC strategy and the material that you're using clinically now? Is that going to be the same material in a pivotal trial and potentially commercially?
Yeah, it's a good question. So, um, the answer is a qualified yes. I mean, we're dealing with such low volumes and such small patient numbers, even in best one. I mean, the smallest true batch that people make is like 50 liters, and then a lot of people make 200-liter batches. We're talking about each patient needing 150 to 300 microliters. And so these batches go a long ways for us. And so we're working with the FDA on what's the most reasonable thing to do, right? I mean, do we really want to be making lots of batches of this stuff, or is it, do we not need as much material? We're also using very well, like this is back to my last point, we're using very well characterized AAVs and promoters, and we're using genes that are the right size to fit into these capsids. And so that's part of it too, is that the manufacturing for a lot of this has been worked out over the years. And so I think when you think about manufacturing, it truly is just manufacturing. We don't spend as much time on experimentation with the chemistry.
Okay, got it. Makes sense. All right. A couple more questions before we reach the end of the session. What is the company's current cash runway, and what do you expect this to carry us through, and what milestones?
It's a good question. Our cash runway is second half of 26, which takes us through the milestones for LCA5 data readout and for best one data readout. And it does not contemplate any contributions from our commercial asset or
Mhm.
from other financing sources that we have.
Okay, makes sense. And the last question that I have that we often like to end our discussions with, what do you think investors are still missing about Opus story?
There's two aspects to our story that are differentiated from other gene therapies. Number one is the quantum of capital it takes to get to what that we think it'll take to get to an approval. So most people think gene therapies take hundreds of millions of dollars and years, like decade. Ours, we think, are going to be much shorter and much less because we're very disciplined on tried and true technologies that we know are going to be effective. And so, or we believe are going to be effective. And so we think that with relatively modest amounts of capital, we can achieve um, approved products. And we think that's really important in these ultra rare populations because the you just don't have that latitude you have with big indications to go out and spend tons of time and tons of money. And so that's very important to us, and it creates a really solid investment thesis for the investor. The second aspect that people sometimes overlook is the whole portfolio we've put together. So we have a commercial asset that is partnered with Viatris, which is going to be producing royalties and milestones for us. And it's we just read out two phase 3 trials for that program in June. And so that's going to add fuel to the fire and is not even contemplated in our runway right now. And so if that truly materializes and actually becomes part of our runway, you may see a substantial change in that. And so it's really there's really a lot of different financial levers that we're pulling between non-dilutive capital, the commercial asset, that are offsetting our equity market needs and allowing the company to achieve a lot with a little.
Mhm. Got it. Excellent. All right. I think with that, we are at the end of our session. Again, George, really appreciate you joining us. Thank you for the time, and I look forward to catching up with you again soon.
Yeah, thank you, Daniil.